So, welcome back. Today in complement learning system, we are learning about alternative pathway. So, in alternative pathway, we have to first understand one thing that like lectin pathway, it is antibody independent, and unlike classical pathway, which requires antibody. So, antigen antibody complex has to be formed for classical. Here, there is no such requirement. So there are various activators of this alternative pathway. Some of them are microbial activator or pathogen activators, and some of them are non-microbial activators. So if microbial activators I talk about, they are like endotoxins, like lipopolysaccharide of gram-negative bacteria, pardon me, picoic acid of gram-positive bacteria, some yeast cells, some parasites like trypanosomes, some virus-infected cells. So these are all direct activators of alternative complement pathway. If I talk about non-alternative pathway, then human antibodies like IgA and IgD can directly activate this. Cobra venom, nephritic factors, which is present in patients of glomerulonephritis, heterologous RBCs from various other species like mice, rabbit, and chicken, dextran sulfate, which is like anion polymer, some pure carbohydrates like agar and inulin can directly activate alternative pathway. So it gets activated directly without antigen antibody complex. It does not require antibody. It is independent of antibody, and it is more or less similar to classical pathway once it reached to the stage of C3 convertase. But just like lectin, we have to first make C3 convertase. So let us start with it. So the first is thing that you have to understand is that we have this free C3 component always there in our serum normally, and this C3 gets spontaneously hydrolyzed by enzymes, and obviously it will form two fragments, C3A and C3B. C3A is a smaller fragment. It has an anaphylactic and chemotactic action, and because it is very small in amount, it does not exert much of the action and it diffuses out. C3B, on the other hand, it's a larger molecule and it attaches directly on the foreign cell surface or the activated molecules. Now, what happens that this C3B usually it is kept in check by factor H and I. But if there is activators, then this activator allow this factor B to bind to this C3B which is coated on pathogen surface. So C3B is continuously generated in small amount in circulation, but it is rapidly destroyed by factor H and I. So it does not get a chance to get deposited on cell surfaces. But if factor B binds to it, then it will protect it from activation from factor H and I. And once it binds to this factor B, it will split this factor B into BA and BB. And BA is again a smaller molecule. And this is a magnesium dependent reaction. And BA will released into the medium, and BB will join the C3 B, and it requires a factor D for this. It is known as C3 proactivator convertase, and it forms C3BBB, and this is actually C3 convertase, but it is unstable. So you require a factor P. P stands for properdin, and properdin stabilizes the C3 convertase, and then rest of the thing is same as classical pathway. So if properdin is deficient, also you will get pyogenic infection and Neisseria infection. So this you have to understand. So alternative pathway is very simple and easy to understand. So now as we have learned all the three pathways, let us look at the summary of them. So classical pathway, it is antibody dependent. It starts with antigen antibody complex. C1QRS binds, split C4 first, then C2, C4B2A complex forms the C3 convertase. In lectin pathway, mannose binding lectin is there, which acts on mannose pathogen on surface, activate MASP enzymes, again cleave C4 and C2 simultaneously, form C3 convertase. Alternative pathway, the C3B, which is constantly generated, it will not get inactivated by factor H and I because it is protected by factor B. The factor B will be split into BA and BB, and BB will be the C3 convertase with help of factor D and propionine factor P. So the C3 convertase will split C3 into 3A and 3B, and 3B will act on C5, 5A and 5B, and 5B will join the uh, membrane attack complex along with 6, 7, 8, and 9. So this is the summary of all the complement pathways. So in next video, we will discuss in detail about various functions of complement pathway.